All right. So, Assalamu alaikum, everyone. My name is Hiba Raheel. I'm an intern. And today I will be giving a PAL session on the reactive oxygen species and the cellular antioxidants. So, this is relatively a simpler lecture, and we should be done pretty soon. If you have any questions, you can send me an email or message me on WhatsApp. I will attach my number and email to the presentation. And of course, you can stop me along the presentation to ask any questions you have. So let's begin by defining what antioxidants are. What reactive oxygen species are. We'll talk about the reactive oxygen species first, and then we'll move on to antioxidants. So reactive oxygen species, or ROS, are basically molecules that contain one or more unpaired electrons. And in a minute, I'll show you what that looks like on an actual picture. So basically, when a molecule has all the electrons, this is what it looks like. Just one second. Okay, so I think you can now see my pointer. So this is what a stable molecule looks like. All the electrons are paired, and this molecule is unreactive, or it's not reactive, okay? Now, all of a sudden, this molecule, let's say, loses an electron. Now, in that case, when it loses an electron, now it has unpaired electrons. So the electrons are not in equal numbers. And now this molecule becomes a free radical, or a reactive oxygen species. And now it has become very unstable, and it is now chemically active. So I hope that with this picture, it's clear what ROS looks like. So what are the different types of ROS? So we have superoxides, hydroxyl radicals, hydroperoxyl radicals, lipid peroxide radical, and nitric oxide. So ROS is a broader term that we use, and then within ROS we have different types of free radicals. We have free radicals, and then we have non-free radicals, right? So ROS is just the broad term that we use. It's like an umbrella term. Now, what about the properties? So what do the free radicals do? So most of the free radicals are derived from oxygen, okay? And in that case, they're called ROS, reactive oxygen species. But there are also some free radicals that are derived from the nitrogens. So in that case, they're known as RNS, or reactive nitrogen species. They can be reactive metabolites or intermediates. So once a free radical is activated, it becomes very, very reactive, and it has a short half-life. So the shorter the half-life is, the more reactive the molecule is. And they generate new radicals. So it's like a chain reaction. So one free radical will go on and generate more free radicals. And this eventually causes damage to the biomolecules, the cells, and the tissues. So production of free radicals is very normal in the human body. And it happens all the time. Actually, there are a lot of uses for the free radicals, and we'll get into those later on in the presentation. So when the production of the free radicals is controlled, it's limited, it's good, right? It's beneficial. But there is a check-in system placed by the body. So there are antioxidants, there's certain enzymes that break down these free radicals and basically neutralize them. So when that is going on, there is like a balance. But as soon as this balance is disrupted and there is production of extra free radicals, and the check-in system is not able to contain the production of the free radicals, what starts happening is we start getting cellular damage, tissue destruction, and basically they start wreaking havoc. So these are some of the different examples of free radicals. I'm pretty sure you're not expected to know all of them. You're just expected to know the more important ones. So if I were you, I'd know what superoxide is, what hydroxyl is. We're going to talk about the peroxynitrate in a second. Nitric oxide is important as well. So peroxynitrate is basically generated from nitric oxide and superoxide. The nitric oxide, or NO, is an important vasodilator. It is very crucial in terms of inflammation when we want the vessels to be dilated for the recruitment of white blood cells and inflammatory markers to the site of injury, the site of inflammation. But however, nitric oxide needs to be removed at a timely manner. If nitric oxide remains, then vasodilation remains, and we have more recruitment of inflammatory markers, and then basically that would just lead to the initiation or the continuation of the process of inflammation. And we also have superoxides. Superoxides are present, hydroxyl and superoxides are both present inside WBCs. They help with the removal of bacteria, and we're going to get into that. Now, for this picture over here on the right side, I want you to focus on the half-lives. I don't want you to memorize it, but I want you to make a point that the shorter the half-life, the more dangerous the reactive oxidative species is. So here, hydroxyl radical has a half-life of nanosecond. Hydroxyl radical is one of the most destructive ROS species if they're not controlled in time. Okay, so now let's get into some of the functions or the possible benefits of the hydroxyl radicals. So first of all, we're going to talk about OH or hydroxyl radical. When we have normally we have iron in our blood, correct? It's present in the hemoglobin, it's present in the blood at all times. So when oxygen and the Fe3+ or ferric iron, when they react together, the superoxide, which is the radical, when it reacts with the ferric oxide, with the ferric iron, it produces oxygen and ferrous. So superoxide or O2, this one right here with an electron that you can see at the top, this is normal. This kind of ROS is normally present in the body, right? It comes from chemical reactions. So it reacts with the ferric iron to produce oxygen and ferrous iron. And then this ferrous iron goes ahead and reacts with H2O2. H2O2 is hydrogen peroxide, or also known as bleach. Again, this is normally present in the body, and when it reacts, it forms the hydroxyl radical or the OH radical. All right. Now, this OH radical is what is the more destructive one. So this is the most destructive free radical out there. We're not as concerned about the superoxide radical here. We're more concerned about the hydroxyl radical. So when we prescribe iron supplements, especially women have a higher tendency to develop iron deficiency anemia. So in this case, what we do is when we prescribe iron supplements, we have to be very careful. So if we prescribe too much of them, it could cause a lot of production of the hydroxyl free radicals, and that could damage tissues, and especially we have to be aware in pregnant females. And what are some agents that produce Fe2+? So we have glutathione, we have ascorbate, we have hydroxyquinones. These are medications. Right, so let's further talk about how the O2 or the superoxide is produced. We spoke about how the hydroxyl radical is produced. Now let's talk about how the superoxide radical is produced. So which pathways produce ROS in our body? So one of the main ones that you need to know are the WBCs. They produce ROS, and the production of the purine nucleotides produce ROS. So WBCs are when they are involved in killing a bacteria, particularly neutrophils, they engulf or they phagocytose a bacteria, and they store that bacteria in an organelle called phagolysosome. Once inside the phagolysosome, there is this very important enzyme, NADPH oxidase, that's present. NADPH oxidase takes this oxygen and it converts it to superoxide. The superoxide is dangerous enough, and it can go ahead and kill the bacteria, damage the bacteria, or it can also be converted into other free radicals like hydroxyl radical and other types of radicals. We're going to talk about that later. So this is just like an overview quickly of what the superoxide does. So this is through one way, through the production of NADPH oxidase. Another way is through xanthine oxidase. So xanthine oxidase is an enzyme that converts oxygen to superoxide when producing purines. And in the process, hypoxanthine is converted to xanthine, and xanthine is then converted to uric acid to produce the superoxide. Now we have superoxide. It's done its job, it's done its damage, whatever we need it for, it's done. Now the question is, how do we get rid of it? How do we get rid of it? We have an enzyme called superoxide dismutase, or SOD. The SOD enzyme is it's basically a very important enzyme that converts this highly reactive superoxide into H2O2. Once it converts it into an H2O2, now H2O2 it can easily be converted into water, because H2O2 itself is still very toxic. It's very damaging. We have to convert it, further neutralize it. It's much easier to convert H2O2 into water through an enzyme called catalase. Catalase takes the H2O2, removes a molecule of oxygen, and makes it water H2O. Another enzyme that we have in place is a GSH peroxidase. So why do we have two enzymes? You'd be wondering, why do we have both these enzymes? Why do we need two enzymes to convert oxygen into superoxide? And why do we need two enzymes to convert H2O2 into H2O? Well, because if one enzyme is deficient or it's not working, you have another reaction, you have another enzyme to compensate, like subhanAllah, because a lot of diseases, we're going to talk about them later, a lot of patients, they have NADPH missing, right? So immediately you think, oh my God, these patients would not be producing superoxide, and then how will they kill bacteria inside the WBCs? Like, how would the WBCs even function and stuff? But subhanAllah, like, we have another alternative pathway. So in case the catalase is not working, we have another enzyme called GSH peroxidase. GSH peroxidase does essentially the same thing, converts H2O2 into H2O. However, it uses a molecule of glutathione. So what it does is that it turns this reduced glutathione, or GSH, into an oxidized glutathione, right? So the GSH becomes GSSG. So it basically converts the reduced one into an oxidized version of glutathione, right? So glutathione, reduced glutathione, or GSH, is actually very, very important for this reaction. Otherwise, GSH peroxidase is not going to work. GSH peroxidase needs GSH in order to convert this reaction, for this reaction to work, basically. Okay, so patients with G6PD deficiency, they get reduced glutathione as supplements. This form, we can give it to them as supplements because they're unable to produce this molecule. We give it to them to protect them from hemolytic anemia. And I think that's pretty much it from this side. All right, so we also need to replenish the stores from the stores of GSH. So we have another enzyme, GSH reductase, which converts the GSSG, or the oxidized glutathione, into a reduced glutathione, and it uses the molecule NADPH to do so. Okay. So this is pretty much summarizing what I explained using that image, that neutrophils produce ROS to kill bacteria in the protective space of the phagolysosome. Phagolysosome is an organelle which contains the phagocytosed bacteria, and it also contains the enzyme NADPH oxidase. NADPH oxidase produces superoxide to kill the phagocytosed bacteria. The production of ROS can also occur at a slower rate wherever there is high concentration of oxygen. So that's very important to remember, such as RBCs. RBCs are also capable of producing ROS. ROS are also a byproduct of the electron transport chain in the mitochondria. And we'll look into that in just a second. I'll show you what that looks like. But here we're just going over how ROS is produced, why it's important. So look, it's produced not just by the WBCs, but also RBCs, also mitochondria. And one very important thing that we need to remember is that when you have a reperfusion injury, ROS are the main thing causing the reperfusion injury. So what do I mean by reperfusion injury? Okay, let's look at here, this slide. So the rate of ROS can increase during a reperfusion injury in a tissue that has been temporarily derived or deprived of oxygen. So let's say you have a heart, and an area of the heart, suddenly the blood vessel to that particular area of the heart is stopped. Okay, there's a blockage in the artery, and the blood supply to like a small area of the heart has stopped. It's halted. Now what? Now the patient has chest pain, you give them medication, you open up the blocked vessel, and so now the blood flows, right? So the time the blood was stopped and the heart tissue was not receiving any blood, it also meant that it was not receiving any oxygen, right? So lack of oxygen, your electron transport chain stops working, ATP levels start increasing, ATP levels start decreasing, NADH levels start increasing because of the deprivation of oxygen, and the deprivation of oxygen of that tissue, right? What has happened when you suddenly the blood supply to the heart, you restore it, you remove the blockade, the blood supply is restored, there's reintroduction of oxygen. Now there's going to be a burst of activity of the ETC, the electron transport chain. And the electron transport chain would be compensating for the lack of ATP in order to basically revive this damaged tissue. So there's going to be an excessive production of ATP, an excessive production of free radicals. And then this is what causes the reperfusion injury. This burst of free radical production is what causes the reperfusion injury. Okay, so to summarize, reperfusion injury is when you have a myocardial infarction, there's a shortage of oxygen, and then the oxygen gets restored, and when it gets restored, it produces free radicals. Okay, so I hope that much is clear. All right. Let's move on. So generation of free radicals, as we already discussed, that 1 to 4% of oxygen is taken up in the body and is converted to free radicals. And then due to the leaks in the electron transport chain in the mitochondria, we have free radicals coming out all the time. We have enzymes in place like xanthine oxidase, aldehyde oxidase, which form superoxide radicals and hydrogen peroxide, and then these radicals go on to the white blood cells, and they perform their function. And macrophages also produce nitric oxide from arginine. All right, now let's further look into the mitochondria and the role of the mitochondria in the production of free radicals, particularly the superoxide. So this is what the mitochondria looks like. Mitochondria, as we've commonly heard, is the powerhouse of the cell. What does that mean? That means that the mitochondria produces ATP. Now, the mitochondria is lined with these proton pumps, also known as complex. So you have complex 1, complex 2, complex 3, complex 4, and so on and so forth. So these complexes, what they do is that they are the major site for the generation of ROS, mainly complex 1. Complex 1 is a proton pump, and the excess delivery of NADH to the complex 1, what do I mean excess delivery? When does that happen? When is NADH excessively delivered? When the electron transport chain goes down in cases of like ischemia, right? So when there's ischemia, the electron transport chain stops producing ATP, you have less ATP and high NADH because NADH is not being used in the electron transport chain. So now you have excess NADH, it goes to this proton pump or complex 1. When it passes through, it results in some electrons being passed from the hydrogen to the oxygen, okay? And now it has an extra electron, and now it is called a superoxide. Now, once you have the superoxide produced, you need to neutralize it, right? How will you neutralize it? By using the enzyme SOD, systolic superoxide dismutase, okay? So this enzyme right over here. So this reaction we were looking at here, this is the reaction that's happening in B. So NADPH to NADH, NADPH takes the oxygen, makes it the superoxide, and SOD then breaks it down into H2O2, then catalase converts it into oxygen, into water. All right, okay. So the mitochondrial electron transport chain of oxidative phosphorylation is the major site of the cellular generation of reactive oxygen species. That is so important to know. Now, in some patients, sod is deficient. We can give them as a protein. We can give it to them as a supplement. That can happen as well. And yeah, so I hope that this slide is clear. If you have any questions, you can drop them in the chat and I'll take a look. Okay, so now let's talk about the WBC. So we spoke about mitochondria. Now we're going to talk about the WBCs and the role of ROS in WBCs. So mainly we're going to look at a neutrophil. So the phagocytic NADPH oxidase, it consumes large quantities of oxygen to generate large amounts of superoxide at a very rapid rate. So the NADPH oxidase, I just showed it to you in the previous slide as well. And we also have inside the neutrophil, we basically have this enzyme. We're going to talk about it again, the NADPH oxidase. It's present inside the neutrophils or polymorphonuclear cells, and they protect our cells or our immune system from bacteria. So this enzyme, very important enzyme, it produces the superoxide radical, and like I mentioned, the superoxide radical can then be converted into other substances like H2O2, or it can directly cause damage to the bacteria. So the H2O2 is then converted by a very important enzyme called myeloperoxidase to HOCl. HOCl is basically it's a powerful oxidant, and it has bactericidal properties, and it's present in bleach. So myeloperoxidase is the enzyme that gives your sputum the green color. When you have a very bad upper respiratory tract infection and you notice that your sputum is getting green. Why does it get green? Because this myeloperoxidase is abundantly expressed in neutrophils, and it has that heme pigment, which is responsible for the green color in the secretions, which are rich with neutrophils. So there's so much, there's so much bacteria that is in your upper respiratory tract. The neutrophils have an excess production of myeloperoxidase, and so when you cough, the sputum actually contains neutrophils containing the myeloperoxidase, and that's the reason why you have green mucus and green pus-like mucus. Okay. So ROS are produced by neutrophils because it's released into the extracellular space. They're fast and have higher output. They're inducible, and they have bactericidal properties. Again, pretty much the same thing that we've been speaking about. I don't think I'm going to go into it one more time because I think I've talked about it enough. Okay. Right, now let's talk about how the free radicals are produced. So we've spoken about how they're produced inside the body, but there are also some outside factors that can influence the production of free radicals, such as the environmental effects like drug metabolism, damage caused by UV or X-rays, cigarettes, alcohol. These are when we say a substance or something is carcinogenic, right? That also means that it has the ability to produce free radicals, damage DNA, damage the genetic composition of a cell. Radiation, UV light, X-rays, gamma rays. That's why we say, you have to limit the amount of X-ray exposure to a patient, because X-rays have the tendency of producing free radicals, which then can contribute into the development of cancer, which we're going to talk about in the upcoming slides. We have chemicals that react to form peroxidase, such as ozone, singlet oxygen. We have chemicals that promote superoxide formation, such as quinones, nitroaromatics, herbicides. It's very important. Patients, you may often hear, patients with, who had a lot of pesticide or herbicide exposure, they develop lung cancer, they develop leukemias. This is one of the causes, because these chemicals have the tendency to produce free radicals. Chemicals that are metabolized to free radicals like phenols and that release iron itself. Remember, iron contributes into the production of H2O2 when it reacts with oxygen. Superoxide, yeah. Okay. Now, what when I say that free radicals, they wreak havoc or they damage excessively, what do I mean by that? What damages are we talking about in particular? So what we're talking about are, for example, proteins. Proteins, they cause oxidation of sulfur hydrate groups and modification of amino acids. So when the free radicals, they damage proteins, they cause modification of the amino acids, which are the basic composition of proteins, right? And they can also damage protein and cause fragmentation, aggregation. When they damage lipids, they can cause cell membrane injuries, because remember, your cell membrane is mainly made up of the lipid molecules, correct? So the cell membrane is highly, highly, highly susceptible to being damaged by the free radicals. So let's take an example. Let's correlate it clinically, right? So we have, let's say, obesity. Obesity is, when people have BMI of more than 34, 35. So it is associated with hyperlipidemia, which means excessive fat. This increased fat metabolism leads to mitochondrial dysfunction and increased ROS generation. And then the mitochondrial dysfunction leads to the activation of stress responses, apoptosis, which then contributes into defective insulin secretion, because these ROS are basically going to start damaging the pancreatic cells or the islet cells that produce insulin. So this is how obesity causes type 2 diabetes. So when lipid peroxidation happens, that means when lipids are damaged by free radicals, it undergoes lipid peroxidation. That can lead to structural alterations, such as alterations in the fluidity and the channels. The membrane-bound signal proteins are going to get damaged. There's going to be an increase in ion permeability. The cell is basically going to lose its wall that's keeping it together. And lipid peroxidation products form cross-links with non-lipid stuff, right? So the lipids are going to start binding to DNA, to proteins. This is going to have a direct cause or direct toxicity of lipid peroxidation. And it leads to disruption in membrane-dependent signaling, DNA damage, mutagenesis, and this is basically setting the background or the platform for the development of cancer. Now, when the superoxides, they damage or they attack carbohydrates, they lead to glycation and an increase in the susceptibility of proteins to be attacked by free radicals. So you see, the free radicals can attack almost every single component of the body. Now, even the nucleic acids. So nucleic acids are what make up the DNA strand. When nucleic acids are damaged, it's going to lead into DNA strands breaking off, fragmentation, eventually leading to cytotoxicity and mutations. Okay. Now let's look at the disease. We looked at the consequences of free radicals on a molecular level. Now let's take a step back, because we don't really care in the hospital setting or in the clinical setting. We really don't care about the small molecular importance of free radicals. We want to know about the big diseases, like, for example, cardiovascular diseases. So free radicals, they oxidize LDL, they cause, they promote atherosclerosis, leading to cardiovascular diseases, also cancers. So free radicals, they cause mutations, cytotoxicity, they cause DNA damage. When DNA gets damaged, it is now mutated, right? Now it's going to multiply and divide abnormally, and now the cells will, because the DNA is damaged, the cells will be dividing abnormally, which leads to the formation of cancers. We also have inflammatory diseases as a result. For example, IBD, if you guys have heard of it, it's like an inflammatory disease of the colon, because the free radicals, they damage extracellular components such as the collagen and the hyaluronic acid, and it produces certain inflammatory diseases. Respiratory diseases, so especially in patients who smoke cigarettes, or not just cigarettes, but also who do shisha or e-cigs, vapes and stuff, they have the ability to produce free radicals, which destroy the endothelium and the lung edema and cause lung edema. And the cigarettes basically contain these excessive amount of chemicals that produce free radicals, which then further produce more free radicals. So like I said, it's a chain reaction. We already briefly spoke about diabetes, and we said that obesity causes extra lipid production, which then causes extra free radical production, which can damage the islet cells or the cells that in the pancreas that produce insulin, right? And male infertility, same concept everywhere. Aging process, it speeds up the aging process. Now, one of the most important diseases related to free radical production is chronic granulomatous disease, or CGD. This is a very important disease for anyone who's interested in taking the Step 1 exam. So you might want to really study this one very well, maybe from first aid or something like that. Because this disease is commonly questioned. So it's basically caused by a genetic deficiency of NADPH oxidase, or G6PD-H deficiency in neutrophils. So we spoke about NADPH oxidase. Let's just go back and quickly look at it one more time before we move on. So here, NADPH oxidase is an enzyme that's present inside neutrophils, the phagol lysosome of the neutrophils. And what it essentially does is that it converts oxygen into a superoxide. So it produces free radicals. And it consumes a molecule of NADPH and converts it to NADP+. And then this superoxide is what will damage the bacteria that has been phagocytosed by the neutrophils. So the bacteria that's been engulfed by the neutrophils will be destroyed by this superoxide, or a free radical. Superoxide is the free radical, right? Then the superoxide has the ability to convert into H2O2, which then can further be converted into an OH, or hydroxyl radical, and HOCl, which both can damage bacteria. So this enzyme is very, very important, because if this is deficient, then this reaction doesn't take place. There's no production of a superoxide radical. So now let's jump back to this slide. So when NADPH oxidase is deficient, then then you don't have the superoxide. It's not produced. And the patient is now susceptible to different types of bacteria, particularly those bacteria that are engulfed and kept inside the phagol lysosome, such as catalase positive organisms. If you don't know what that is, that's fine, but just for the sake of completion, I'm just going to quickly talk about these bacteria. So we have Staph aureus. It's a very, very common and very, very nasty bug. It's very common. It causes infections, abscess formation. We have Klebsiella, E. coli. E. coli causes UTIs, stomach bugs, can cause diarrhea. Candida, that's fungus, can cause oral thrush or vaginal candidosis. Then we have Aspergillus. So these are all enzymes that are these are all bacteria that are heavily dependent on NADPH oxidase, the function of NADPH oxidase. If this is not functioning, then these bacteria are going to remain inside the phagol lysosomes. And eventually what is going to happen is that the neutrophil is not able to kill these bacteria efficiently, right? And it's there, it's inside the neutrophil, but they're not dying. So the immune system detects it, and it kind of walls off this neutrophil. It builds like a wall, or like a granuloma around it, to contain the bacteria, the neutrophil containing bacteria inside that walled off structure. So this is what it looks like, really. This is a granuloma. I mean, this is what you can see from the outside. As you can see, it's excessive. It's just a walled off structure, basically. This is probably, it contains pus, it contains neutrophils, contains all nasty stuff. Okay, now one last thing before we talk about the balance or the removal of superoxidase, the free radicals. So there are certain medications that cause the production of the free radicals. So for example, paracetamol toxicity. Paracetamol toxicity leads to oxidative stress. And remember, we said whenever there's oxidative stress, that means that there is production of free radicals. And the breakdown of the paracetamol produces the process or the breakdown when we ingest a heavy amount of paracetamol, the degradation pathway, it leads to free radical intermediates, which can then damage the kidney and liver. And so sometimes people take paracetamol with alcohol, right? Um, like for example, commonly you would see in movies or TV shows, they overdose paracetamol with alcohol, and the intent is because the combination of alcohol with paracetamol, it causes more damage to the kidney and liver. Okay. Now, this was the picture that I was talking about at the very beginning, where I said that free radicals are neutralized by substances called the antioxidants. So there's the system, and it's like a balance, right? You have production of ROS, which I said is very important. We need ROS in our body. They're crucial substances. So we have ROS, but at the same time, we have these like check-in systems in place that neutralize the ROS when the job is done. So whenever there is, let's say this side gets heavier, or like there's more production of reactive oxygen species and lipid peroxide, what happens is it leads to oxidative stress, right? So major diseases that can happen. Parkinson disease, I'm sure a lot of you have heard of it. High blood pressure, arterial sclerosis, myocardial infarction. We spoke about the reperfusion injury, dementia, cataracts. So essentially all the inflammatory process, all the diseases, the cancer formation, this is the basic. This is where it starts. The excessive production of ROS. This is where it all begins, when the antioxidative system does not work or has stopped working or it's insufficient. And when this outweighs this one, this leads to the basis, the starting of multiple diseases in the body. This is why it's very important. This topic is very important for us as doctors. Okay, now let's talk about these defense systems that we've been mentioning so much. Now, defenses against ROS are basically, we have two important enzymes that we've previously mentioned, superoxide dismutase, or SOD, and catalase, right? So our RBCs, they produce superoxides because there is spontaneous dissociation of oxygen from hemoglobin. Hemoglobin reaches, or blood reaches the tissues and the cells, and then immediately the cells want this oxygen. When oxygen dissociates, it produces superoxides. And so we have, so the products in the RBCs are methemoglobin and superoxide, right? Because of the dissociation of this oxygen. So we have these very important enzyme catalase present inside the RBCs to neutralize the superoxide. And so the deoxidification of superoxide can happen with superoxide dismutase, catalase, glutathione peroxidase, the GSH peroxidase that we spoke about in the slides previously. That's the one. Vitamin E, vitamin C. That's why, they say orange juice is really good. It's an antioxidant. I'm sure like the girls would know, like vitamin C, they say put it on your skin. It's an antioxidant. It resolves hyperpigmentation, blah, blah, blah. It's because of this, because it neutralizes the ROS. And then the low levels of these detoxifying substances can obviously lead to hemolysis of RBCs, and basically it's like, all of these diseases. Yeah, so the antioxidants, they have anti-inflammatory agents, they have nitric oxide synthase, like let's say the body is not able to efficiently work or have its defense system work. So we can also help. We have anti-inflammatory agents. Remember, like paracetamol is an anti-inflammatory, ibuprofen is one of them. They all resolve these ROS. We have nitric oxygen synthase inhibitors. Then we have some metachelators like lactoferrin, transferrin, for iron overdose or something. We have NADPH oxidase inhibitors and xanthine oxidase inhibitors. Xanthine oxidase inhibitors are given in it's a chemotherapeutic drug. It's given in cancer because it destroys the free radicals. Just a side note, insulin also has anti-inflammatory effects, and it can be used as an antioxidant. And yeah. Again, this is just a brief overview of what we've so far talked about. I already mentioned that ROS are very important for RBCs, and patients with G6PD deficiency, they have hemolysis or abnormal breakdown of the RBCs. They cannot produce enough NADPH. As a result, NADPH is lacking, and they cannot neutralize the H2O2. So they're unable to neutralize this H2O2. H2O2 will then go on and damage the cells. So we have this like formation of Heinz bodies, which are basically aggregates of hemoglobin, because this H2O2 will go, it will cause hemoglobin denaturation and membrane damage. And then when hemoglobin denaturates, it forms these collections, also known as Heinz body. So this is a classic image of someone with G6PD deficiency. And with that, we are done. Please scan this barcode and give me your feedback. How can I do better next time? What did you like about this presentation? How can I improve the next time? And if you have any questions or if you have any concerns, you can email me or WhatsApp me. I will attach my number to this presentation, my email and my number. And thank you so much.